Okay, so the next is the cloning vectors. Cloning vectors. Vectors are DNA molecules that carry a foreign DNA segment and replicate inside the host cell. They're also known as the vehicle DNA. Vectors. These are the DNA molecules that carry a foreign DNA segment and replicate inside the cell. DNA molecules that carry foreign DNA. They are known as the vehicle DNA or carrier DNA. They are engineered in such a way that they help easy leaking of foreign DNA and selection of the recombinants from non-recombinants. Vector may be plasmids, bacteriophages, cosmids, yeast, artificial chromosomes. So vectors can be plasmids, bacteriophages, cosmids, yeast, artificial chromosomes, YACs. Bacterial artificial chromosomes, BACs. Also, bacterial artificial. Okay, they and the transposons, etc. The most commonly used vectors are the. So these are all the vectors. Most commonly used vectors are the plasmid and the bacteriophage. Bacteriophage. These are the most commonly used vectors. Commonly used vectors. The following are the features that are required to facilitate cloning into a vector. Following feature facilitate cloning into a vector. First is the ori. It is the origin of replication. This is a sequence from where replication starts, and any piece of DNA, when linked to this sequence, can be made to replicate within the host cells. This is a sequence from where replication starts from ori. This sequence is also responsible for controlling the copy number of the also control the copy number of linked DNA. So if one wants to recover many copies of the target DNA, it should be cloned in a vector whose origin supports high copy number. Next feature is the selectable marker. The vector requires a selectable marker which helps in identifying and eliminating non-transformants and selectively permitting the growth of transformants. Sin identifying and eliminating non-transformants and selectively permitting the growth of the transformants. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium. Procedure through which a piece of DNA is introduced in a host bacterium. Normally, the genes encoding resistance to antibiotics such as ampicillin, chloram, chloramphenicol, tetracycline, or the caramycin, etc., are considered useful selectable markers for the E. coli. Normally, the E. coli does not carry resistance to any of these antibiotics. The genes encoding resistant to antibiotics. Genes encoding resistance to antibiotics. Ampicillin, chloramphenicol, tetracycline, or caramycin. These are the genes which contain resistance against antibiotics. Third is the cloning sites or the recognition. The vector must also have at least one unique restriction endonuclease recognition site to enable foreign DNA to be inserted into the vector for the generation of a recombinant DNA molecule. The vector must also have at least one unique restriction endonuclease recognition site to enable foreign DNA to be inserted into the vector for the generation of a rDNA molecule. Presence of a unique restriction site allows a particular restriction enzyme to cut the vector only at that site. Most of the commonly used vectors contain unique recognition sites for several restriction enzymes in a small region of the DNA. Most of the commonly used vectors contain unique recognition sites for several restriction enzymes in a small region of DNA, which is referred to as the polylinker or multiple cloning site. MCS. The presence of restriction sites within the markers tetracycline and the ampicillin permits an easy selection for cells transformed with the recombinant PBR322. Selection of recombinants due to inactivation of the antibiotics is a burdensome procedure because it requires simultaneous plating on two plates having different antibiotics. So, alternative selective markers have been developed which differentiate recombinants from non recombinants on the basis of their ability to produce color in the presence of chromogenic genic substrate. The inactivation of a gene by insertion of a DNA sequence within the coding sequence of an enzyme, beta galactosidase. This results into inactivation of the gene for the synthesis of this enzyme, and the colonies do not produce any color. These colonies are marked as the recombinant colonies. Vectors for cloning genes in the plants and animals. Agrobacterium tumefaciens, a pathogen of the several dicot plants, is able to deliver a piece of DNA. Agrobacterium tumefaciens is a pathogen of several dicot plants, which is able to deliver a piece of DNA known as the tDNA to transform normal plant cells into a tumor and direct these tumor cells to produce the chemicals required by the pathogen. Pathogen of the several pathogen of the dicot plants. Okay, and the it it is able to deliver a piece of DNA known as a tDNA, which is able to transfer normal plant cells into tumor and direct these tumor cells to produce the chemicals required by the pathogen. Similarly, retroviruses in animals have the ability to transform normal cells into cancerous cells. Retroviruses also have ability normal cell into cancerous cells. The tumor inducing plasmid of the Agrobacterium tumefaciens has now been modified into a cloning vector which is no more pathogenic to the plants but is still able to use a mechanism to deliver genes of interest into a variety of the plants. Retrovirus have been disarmed and are now used to deliver desirable genes into the animal cells. PBR 3022 was the first artificial cloning vector constructed in first artificial cloning vector vector constructed in 1977 by the Bolivar and Rodriguez. Shuttle vectors can exist in both the eukaryotic cell and the E. coli. Most of the eukaryotic vectors are shuttle vectors. Competent host for transformation with the recombinant DNA. Competent host for transformation with the recombinant DNA. Competent host is essential for the transformation with the recombinant DNA. Many kinds of the host cells, including E. coli, yeast, animal, and the plant cells, are available for the genetic engineering. DNA is a hydrophilic molecule. It cannot pass through the membrane, so bacterial cells must be made capable to take up DNA. DNA is hydrophilic molecule. So bacterial cells must be capable to take up the DNA. This is done by treating them with a specific concentration of a divalent cation. Divalent cation, such as calcium ion, which increases the efficiency with which the DNA enters the bacterium through pores in its cell wall. Example, calcium ion. It cannot pass through the membranes. Recombinant DNA can then be forced into such cells by incubating the cells with rDNA on ice and followed by heat shock and then putting them back on ice. In the eukaryotic cell, the term transformation is replaced by the term transfection. In eukaryotes, transformation is transfection. Alright. Different vectorless gene transfer methods have been used to introduce the rDNA into the recipient cells of animals. Recipient cells of animals. Some of the important methods are the gene gun method. In this method, cells are bombarded with the high velocity microparticles of gold or tungsten coated with the DNA. This method is suitable for plants. Important crop plants like the wheat, rice, maize have now been transformed by this method. Cells are bombarded with the high velocity microparticles. Microinjection. In this method, the rDNA is directly injected into the nucleus of an animal cell. Directly injected nucleus into animal cell using capillary glass. Using capillary glass, micropipette, or the microsyringe. Electroporation is the formation of the temporary pores in the plasma membrane of the host cells by using lysozyme or calcium chloride. Certain chemicals such as the polyethylene glycol help foreign DNA to enter the host cell. Then the process of rDNA technology. The rDNA technology involves the following steps in the sequence. That is the isolation of genetic material, DNA. Next is the cutting of DNA at specific locations. Third is the amplification of gene of interest using PCR. Using PCR. Fourth is the insertion of rDNA into the host cell. Then the fifth is obtaining the foreign gene product. Sixth is the downstream processing. So, the first step, which is the isolation of the genetic material, the removal of the plasmid or the genomic DNA from the cell is termed isolation. In order to cut the DNA with the restriction enzyme, it needs to be in pure form, free from the other macromolecules. Isolation usually involves the Isolation usually involves the breaking of the cell's membrane and possibly also cell wall. DNA is obtained by treating the bacterial cells, plant or animal tissues with enzymes such as a lysozyme, cellulase, or the chitinase. Genes are located on the long molecules of the DNA intertwined with the proteins such as histones. The RNA can be removed by the treatment with the ribonuclease, whereas the proteins can be removed by the treatment with the protease. Other molecules can be removed by the appropriate treatments, and purified DNA ultimately precipitates out after the addition of the chilled ethanol. Next is the cutting of the DNA at specific locations. Under optimal conditions, restriction and enzyme digestions are performed by incubating purified DNA molecules with the restriction enzyme for that specific enzyme. Agarose gel electrophoresis is used to check the progression of a restriction enzyme digestion. After having cut the source After having cut the source DNA as well as the vector DNA with a specific restriction enzyme, the cut out gene of the interest from the source DNA and the cut vector are mixed with the lyase is added. This results in the formation of the recombinant DNA. The next is the amplification of gene of interest using PCR. The polymerase chain reaction techniques was invented by the Carey Mullis. Polymerase chain reaction. Carey Mullis in 1985. PCR is best defined as the DNA replication in vitro. In this reaction, multiple copies of the gene of interest is synthesized in vitro using two sets of primes and the enzyme DNA polymerase. The enzyme extends the primers using the nucleotides provided in the reaction and the genomic DNA as a template. The process of replication of the DNA is repeated many times and the segment of the DNA can be amplified to approximately billion times. Repeated amplification is achieved by the use of the thermostable tag DNA polymerase, which is isolated from the thermophilic bacterium Thermus aquaticus. It remained active during the high temperature induced degeneration of the double stranded DNA. A single PCR amplification cycle involves three basic steps. That is the denaturation, that is DNA is heated to a high temperature, usually 90 to 98 degrees Celsius. Second is the primer annealing, which is the two oligonucleotide primers annealed to each of the single stranded template DNA. Temperature usually 40 to 60 degrees Celsius. And third is the extension. That is the tag DNA polymerase synthesize the DNA region between the primers, optimum temperature 72 degrees Celsius. Some of the areas of the application of the PCR are diagnosis of the pathogen and the specific mutation, DNA fingerprinting, prenatal diagnosis, gene therapy, etc. Next was the insertion of the rDNA into the host. There are several methods of introducing the ligated DNA into recipient cells. Recipient cells, after making them competent to receive, take up DNA present in its surroundings. The vector DNA and the alien DNA carrying a gene of interest are cut by the same restriction endonuclease to produce the complementary sticky ends, which are joined by the DNA ligase to produce the rDNA. If a rDNA bearing gene for resistance to an antibiotic, example ampicillin, is transferred into the E. coli cells, the host cell become transformed into the ampicillin resistant cells. If we spread the transformed cells on the agar plates containing ampicillin, only transformants will grow, untransformed recipient cells will die. Since due to the ampicillin resistance gene, one is able to select a transformed cell in the presence of the ampicillin. The ampicillin resistance genes in this case is called the selectable marker. Obtaining the foreign gene product. Most of the rDNA technologies are aimed to produce a desirable protein. The foreign gene gets expressed under appropriate conditions. After the cloning of the gene of interest, one has to maintain the optimum conditions to induce the expression of the target gene and consider producing it on large scale. If any protein encoding gene is expressed in a heterologous host, it is known as a recombinant protein. The cells can also be multiplied in a continuous cultural system where the used medium is passed out from the one side and the fresh medium is added from the other side of to maintain the cells in their physiologically most active log or the exponential phase. This type of culturing method produces a larger biomass leading to higher yields of the desired protein. Bioreactors are considered as a vessel in which raw materials are biologically converted into specific products by microbes. Plants and animals or the human cells. A bioreactor provides the optimal condition for obtaining the desired products by providing optimal growth conditions such as temperature, pH, substrate, vitamin, oxygen, and salt. The most commonly used bioreactors are of the stirring type. Stirring type bioreactors are of the further two type, which is a simple stir tank bioreactor or the sparged stir. The sparged stirred. A stirred tank reactor is usually cylindrical or with a curved base to facilitate the mixing of the reactor contents. The stirrer facilitates even mixing and oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. The bioreactor has an agitator system, an oxygen delivery system, agitator, and the oxygen delivery system, and a foam control system. A temperature control system, pH control system, sampling ports so that the small volumes of the culture can be withdrawn periodically. Last is the downstream processing. After completion of the biosynthetic stage, the product has to be subjected through a series of processes before it is ready for marketing. As a finished product, the process includes the separation and the purification. which are collectively referred to as the downstream processing. The products has to be formulated The product has to be formulated with the suitable preservative. Such formulation has to undergo through clinical trials as in the case of drugs. Strict quality control testing for each product is also required. Downstream processing and the quality control testing varies from product to product.